but we're going to kind of do a small review here about some of the components to the immune system. Right? And so let's start off on the smallest components, the cells. And so that's going to be our white blood cells, also known as our leukocytes. Keep in mind, all the foreign blood elements of your blood are going to be produced in the red bone marrow, not the yellow bone marrow, but the red bone marrow. So our two white blood cell classes are agranulocytes and granulocytes. And so we'll start with the latter first. Let's start off with the granulocytes. That's any cell, any white blood cell that has Phil at the end of their name. So neutrophils, which are the most common type of white blood cell, right? Then we have our eosinophils and our basophils. So those are going to be the ones that we're going to discuss. And then we have our other cells, the monocytes and our lymphocytes. And so the monocytes are our blood not blood formed, but our blood macrophages, essentially. And then we have our lymphocytes, which include, right, our natural killer cells, which is a cool cell, our T lymphocytes and our B lymphocytes. So we're going to kind of go through all of this here, just kind of like a review here. And we talked a little bit about some of these structures in lab on chapter 21, but we'll discuss it here, all right, why isn't this working? We'll talk about it here a little bit in lecture. What the heck? Anyways, okay. So let's start talking about where these immune cells are going to be located. And then this is something that I mentioned before, but I'm going to highlight it here for you so you don't forget it, right? Most of your white blood cells are in your tissues, not your blood. A lot of folks think that most of the white blood cells are in the blood. They are not. So we're going to be talking about some of these tissues, where they're housed. For example, our secondary lymphatic structures. And we discussed some of those before, right? So our secondary lymphatic structures, if we go through the list here, right, you'll see T and B lymphocytes, macrophages, which are the monocytes that have migrated out of the blood into our tissues here. Dendritic cells, we learned about those in chapter 6, all right? They were sitting in the stratum spinosum there, and so they played a big role in detecting early cancerous, and sometimes when pathogens have invaded through the epidermis, they got to get by the dendritic cells. And then we'll talk about some of our natural killer cells here that just hang out in the lymph nodes, and their name tells you exactly what they do, all right? They go around and just kill stuff. All right, and then we discussed a little bit when we were talking about chapter 21, the spleen and tonsils. And then we've got our MALT structures, the mucosal, right, associated lymphatic tissue, right, those pair patches that we discussed that are reside in the ileum. I think you all remember I did mention that the majority of your immune system is located along your digestive tract because you are taking things that were external, right, and that could be pathogen-ridden. You're bringing them into your body, you're chewing it up, you're swallowing it, and you're going to try to absorb that material through your digestive gut lining. And so those pathogens, some are pretty sturdy and can handle, all right, living in some of those structures, and then can migrate through, all right, the epithelium, and then your body has to combat it. Well, guess what? We got these MALT structures here that will help with that. And then as we go through these different chapters in this course, we're going to see, right, that our macrophages, those are the monocytes that take up residence in some of our other tissues, right, are going to be specific, right, to certain organ systems. Right, for example, right, you have macrophages that like to live in your lungs. And if you think about it, especially if you live in an urban area, I don't really consider Greenville, too urban, but it is, downtown is considered urban. You're going to have an increase in particulate matter in the atmosphere. And so, especially when we were dealing with COVID, all right, an airborne illness there, when you're inhaling these pathogens and these viral particles, right, we need a first line of defense. And so these macrophages that hang out here in some of those tissues are going to handle that, all right? And you'll see kind of the role that the macrophages play, right? Again, they're like I guess if you follow baseball, they're the utility player of our immune system. They do a lot of different things. They clean up a lot of messes, and they can initiate the immune response on a lot of different occasions there. All right, so where can we see some of these immune cells here? in the epidermis there, mentioned that, and mucosal membranes. Specifically, we've got a lot of immune structures in the mucosal membranes there, right? Antibodies. Antibodies are these proteins that are produced by plasma cells. Plasma cells at one point used to be B lymphocytes. They differentiated into plasma cells and they made antibodies. Right? Antibody A, we'll discuss this later on in this chapter, that there's different types of antibodies, but antibody A loves to hang out in the mucosal membrane. So your mucosal membrane is going to line a lot of entrances and exits to your body. And that's the purpose of these mucosal membranes is for protection. And so when you're breathing in, for example, in your nasal cavity, you've got that pseudostratified ciliated columnar epithelium. Say that real fast, all right? And so the goblet cells, amongst some of the other types of glands, are going to produce a mucus coating throughout your nasal cavity. And so in that mucus, you're going to have these IgAs, all right, the immunoglobulin A, the antibodies there, that are going to come in contact with some of these viruses, pathogens, and they're going to start to initiate the immune system. And so we'll talk about how all that works there. So kind of keep that in mind when we're discussing some of the locations there. Connective tissue, which is usually deeper to these two other structures here, the epithelial layers and the mucosal membrane there, okay? Connective tissue, we learned about, very important cell right here, our mast cells. Right? And our mast cells, along with a lot of other types of immune cells, like macrophages, for example, and our connective tissue, if you remember in chapter 5, we learned about wandering cells. And those are the cells that can move around through the connective tissue. And then you have what's called resident cells, and they're just stuck there, right? So macrophages play both roles. So they can move around through the connective tissue, or they're just going to be embedded into that connective tissue. You'll have dendritic cells, right? And then these mast cells here, right, are also going to play a role because they're going to be close to those small blood vessels. Why is that important? Do you remember what mast cells secrete? There's two important chemicals that mast cells secrete. That's right, you nailed one. So histamine, which is what? What does it do? Do y'all remember what it does? Well, it's part of the inflammatory response, but it makes your blood vessels leaky. All right, so it causes gaps to appear in between the endothelium that lines your small blood vessels, so stuff in the blood can leak out into the tissues, which is good because white blood cells that are in your blood can then escape, and we'll talk about that process. They'll escape out of your blood and get into your tissue and then handle an infection that way. And it also releases heparin, the two H's, histamine and heparin, blood thinner, right? But we consider it a blood thinner, but it's an anticoagulant. Okay, so it's going to prevent, because once we get gaps in the blood vessels, immediately what does your body want to do? It wants to clot. It wants to block that off so you're not leaking stuff out. So if we release the histamine, it prevents the clotting so we can leak stuff out appropriately. It's kind of a cool system. So mast cells are going to stay in the tissues, but in close proximity to your blood vessels. Do you remember the granulocyte that is pretty much similar to the mast cell? You nailed it. Yeah, basophil. Yep, basophil. It does the same thing, right? But it's in the blood. So you've got a cell that's in tissues that does what it does. And then you've got, all right, cells that are in your blood. So it's on both sides of the endothelium there that will help. All right, so again, we're going to see, all right, a lot of these mast cells located throughout, especially, come on, y'all. For those of you that suffer during allergy season, you're going to see lots of mast cells in the respiratory system. Gives you a runny nose all the time. Some of you might even take antihistamines, right, to help curtail your allergy responses there, right? So we're going to see mast cells, and their job is to make your blood vessels leaky so your white blood cells can kind of go about their business and escape out of the bloodstream and into the tissues and kind of do their thing. So this picture here just showed you. Here we have all of our white blood cells that are in circulation. Keep in mind, most are in your tissues. And then they'll migrate out of circulation and get into some of these tissues, or they've already taken residence in some of those tissues, right? And they're just going to hang out there until they encounter some sort of pathogen. All right, so one of the things you're going to learn in this chapter is a lot of our immune system operates on chemical signals. So cells, pathogens are going to release chemicals. And so these chemicals, we're going to give them a name. We call them cytokines. They're going to help to regulate the immune response, right? So when we talk about cytokines, they're a protein. And so they're going to regulate this immune response, and we'll talk about how that is. But when we talk about the immune system, there's actually two parts to it. Some people say three, but we're going to go with the two because our book talks about it. You have what's called the innate immune system and our adaptive immune system there, right? And we'll spend today, at least in this first class period, we'll definitely talk about innate and everything that goes along with it. And then we'll talk about adaptive and cell-mediated responses and all that. All right, so here's what happens, right? You're going to have these chemicals, these cytokines here, that are going to be released from a certain cell. It could be a white blood cell, right? And in some cases, it could be an unhealthy cell that's been infected, maybe, for example. All right, but point being is, you're going to get this chemical, the cytokine, is going to be released from a cell, and it then is going to bind onto a target cell. And we'll discuss what all the different things that are going to happen here, right? So going back to chapter 17, those effects can be autocrine, which means the cell itself releases that cytokine that acts upon the cell that released it. We've got paracrine. Remember that? That's going to be a cell that releases, all right, the chemical or the cytokine that's going to affect all the neighboring cells. And then, of course, we have endocrine, which means that cytokine gets into the bloodstream, and then it's going to circulate throughout the entire body, right? And so it can go anywhere, right? These chemicals will have a short half-life. What does that mean? Pardon? Take a guess. I won't make fun of you for very long. Just kidding. Short half-life means it doesn't last long. It breaks down quickly, okay? So if you have a long half-life, you'll last a little bit longer. If you have a short half-life, right, you won't last very long. And by design, because you don't want these chemicals just sticking around forever, because then we can start to, increase the likelihood or the possibility of an autoimmune response. And take it from somebody who knows. You don't want to be messing around with autoimmune responses, all right? Because they suck, all right? But point being is, it's a good thing that we have our short half-life. So what are some of these effects here? All right, well, one, they can act like a signal flare. And so they can actually just set up and shoot out, this message like, hey, there's something going on here. And so they can act as signaling cells to draw attention to themselves, right? Next one is controlling development and behavior of immune cells. This is where we're going to really get into, when we get into cell-mediated immunity, we're going to learn about how certain lymphocytes, like our T lymphocytes and our plasma cells, we're going to see how certain cells are going to react when you've heard me say, well, this occurrence happens and it could initiate the immune response. Now we're going to see what that immune response is, all right, for specific cells. And we'll discuss all that, right? Today, we'll talk about how it's going to regulate the inflammatory response, because believe it or not, inflammation is a good thing to have initially when you have some sort of damage to tissue or a compromised tissue. And then finally, let's face it, it boils down to destroying cells if you have, like, for example, a tumor cell. All right, if a cancerous cell occurs, an unhealthy, unwanted cell, or an unhealthy cell that has been infected by a virus, because that's what viruses like to do, right? If you're taking some of you guys are taking micro, right? Christian's not even here. He's missing all the good stuff, all right? So you're going to see that viruses love to just invade cells, convert all the internal machinery over to virus-producing machinery, and just pump out viruses like a factory. So we got to, it's again, going back to, what's it called? What's his name? The Art of War by Sun Tzu, all right? You have to destroy the resources of your enemy. So the enemy in this situation, this is what immune response is. It's like warfare, all right? Enemy here in this situation is that cell that's pumping out viruses. We got to take it out. Okay, so we'll talk about all that. And who would have thought that cytokines would have played a role in that? Okay, so let's talk about our two types of immunity, adaptive and innate. We're going to start off with innate, right? But we're going to talk about some of the differences here. Is it bad to be drinking coffee at 6 o'clock at night? Okay. Well, you're drinking an energy drink, aren't you?
No.
Looks like
it's a protein iced coffee.
Oh, high protein iced coffee. Oh.
It's, well, It says shake gently and crack slowly, and I decided.
You did not do that. Okay. All right. Well, it looks like a truly, so I wasn't sure what you were bringing into class. All right. So let's talk about, all right, how, when we're discussing the immunity, what's different? All right, how? One is the cells that are involved in each response. Okay, you're going to see that, all right, some cells are going to be specific for innate immunity, some cells are going to be specific for adaptive immunity. How they respond, okay, how specific they're going to respond to that pathogen, let's say, right? And then how they're going to take care of business. But how do I want to say that? Mechanisms of eliminating harmful substances. How are they going to take out, right, those pathogens? And then how long does it take? How long does it take for that immune response to occur, and how long will it last for? All right, well, so we'll talk about these. Right? The whole point is, though, that all of these different types of immunity, right, work together for the overall well-being of the organism, us. Okay, their job is to keep us from getting sick or dead. All right, their job is to protect us from pathogens that have invaded. So that's what we're going to discuss here. So let's look at the two types of immunity here real briefly. We're going to get into more detail here. All right, so you'll see we have innate immunity and then adaptive immunity. So innate is you get that at birth. So you're born with that. And when we look at some of the examples, like for example, I'm going to skip to here. Innate immunity is your skin. That's one type of innate immunity. It acts as a barrier, right? Wouldn't you say? Keeps a lot of pathogens. It keeps, I know I keep saying pathogens, bacteria, viruses from getting into the more sensitive tissues, protects you from the sun, right? Your skin protects you from the ultraviolet radiation there, right? Keeps you warm, okay? Cools you down, hopefully, all right? So you're going to see, right? The big thing with innate immunity is how it works is that it's non-specific. It keeps everything out, right? For example, one of the characteristics of your skin is that it's water resistant. Notice I said resistant and not waterproof, right? It'll keep water out. It'll keep pathogens out for the most part. So it can't differentiate. It can't let one thing in and not others. So it's very non-specific here because it is going to experience a variety of different stresses, challenges, but it has one job. Be a barrier, like the walls of your house, all right? Keep out intruders, hopefully, all right? Your skin should keep out, all right, everything. All right? And then we'll also see that with our mucosal membranes here. And then we'll talk about the different types of cells that are involved in here. The nice thing about innate immunity is it's pretty much immediate. Okay, we don't have to wait around. When we get to adaptive immunity, it's a different story. The nice thing about adaptive immunity is it's specific. It's specific. So once you've encountered, for example, a pathogen like chickenpox. Now, I know they don't really do, but when I was growing up, we didn't have a chickenpox vaccine. So I'm dating myself a little bit. We didn't have a chickenpox vaccine. But what we did have was chickenpox parties, right? And so and I know that some of you are laughing because you know what I'm talking about, all right? If a kid in the neighborhood got chickenpox, all the parents in that neighborhood, all right, would try to get their kids to play with the sick kid, right? When does that ever happen anymore, right? So we'd have them go play with the sick kid and they get chickenpox, and then they would get immunity from chickenpox because they already had it. And so we'll discuss how that occurs. So they basically acquired this immunity, and now they are going to be protected against that specific pathogen. So this is when we're going to utilize our lymphocyte cells, the T and B lymphocytes, which means that specific T lymphocytes and B lymphocytes are going to be, how do I want to say this? They're going to be specific for, I'm using specific a lot in this sentence here. They're going to be specific for a specific antigen. What does that mean? An antigen is a foreign protein or a sugar. Okay, it's foreign. And so it could be a virus, pathogen, or whatever, or bacteria. And so what happens is when you encounter, and I'm trying not to go into detail on this because we're going to go into a lot of detail here soon. When you get exposed to that chickenpox or even COVID, right? The next time that you encounter it, your response will be a lot shorter to actually get rid of that pathogen, or you might not even know you're sick because your immune response is much larger and much more capable of taking out that pathogen before you become symptomatic. I mean, a lot of times, what are some of the symptoms that you get that tell you that you're sick? Fever, tiredness, yeah, you could be sweating, malaise, all sorts of different things. So you might not even experience any of those when you're exposed to a pathogen that you've already had an immune response to. And that's the nice thing about this. So for some of these cases, right, instead of waiting weeks possibly to get over whatever illness you have, if you are exposed to it again, it can be days, hours even, right? So we'll talk about how this all works. But here's what we're going to say for adaptive immunity. A particular cell responds to one specific foreign substance, but not another. Which means that certain cells are specific for chickenpox. Certain cells are specific for COVID. Certain cells are going to be specific for, all right, viral meningitis. So it's going to be specific. So when we get to adaptive immunity, innate, not so much. All right, so I would say this is figure 22.2. Look at this table, this chart here, and start with this when you're starting to understand the immune system. You'll notice on the left side of our screen here, right, we got our innate immune system. On the right side of the screen, we got the adaptive. So pretty much, hopefully we'll get through at least most of the innate immune system this first class here. And then we'll finish it up in the second class and we'll start working away on the adaptive immune system here. All right, so you're going to see as we go through, right, when we're talking about the different components here for the innate immune system, right, we've got our membranes, the cutaneous and mucosal membranes, right? Those are our barriers. And then we'll get into our non-specific internal defenses. And you'll see we have cells, chemicals like interferon and whatnot, and then physiological responses like inflammation and fever. So we're going to discuss that and why it's a benefit. I personally believe that fever is a good thing. The low-grade fever, high-grade fever, 104, 106, not so good. All right, we don't want to mess around with that. All right, so let's go into a little bit more detail here, talking about innate immunity. All right, so let's look at some of the characteristics. Like I said before, prevents entry of potentially harmful substances. Yes, it is non-specific. Okay, that could be good because if it's non-specific, that means it's going to prevent entry. For example, if we're talking about our membranes there, the cutaneous and mucosal, it's going to prevent entry to everything. Okay, we're shutting everything down here. So we have two lines of defense. The first line of defense are the barriers, skin and mucosal membrane. The second line of defense is going to be the stuff that goes on. So say, all right, a pathogen manages to make it past your skin and your mucosal membranes. It's a really persistent little bugger. And so it gets through, and then it gets down into the deeper tissues of your body, right? Through the dermis and down into the connective tissue below that. Right. So now we're going to talk about what that pathogen can experience or encounter, right, once it's gotten past our first line of defense. And there's a slew. And you've learned this kind of, because if you remember, deep to our skin is a layer of connective tissue. And so a lot of the cells in our connective tissue are white blood cells. Specifically, these guys, neutrophils, are the most abundant type of white blood cell. Right? They do so much. They're awesome cells. And then one of my favorites, the macrophages. Look at all these different cells here that you'll encounter. Right? Our dendritic cells, natural killer cells. Eosinophils and basophils can be, all right, in the tissue if they've migrated out of the bloodstream. Okay, normally we'll find them in the bloodstream, but there's an ability that they have that they can leave. And then we're going to talk about how, all right, some of these tissues, these cells are going to produce, all right, those chemicals that I was discussing earlier, right, but interferon and then complement. And then finally, we'll talk about inflammation and fever. So you have a lot of tools at your disposal when you're initiating the immune response. The nice thing is I love the fact that my skin keeps out so much stuff. Is anyone, and when I took my micro class, we had, you guys taking micro, have you swabbed anything yet and cultured anything? You have? Did anyone in your classes swab their body, like their skin or anything? Did they find some interesting, yeah, it's crazy. It's crazy what you'll find just living on your skin. All right. And you'd be surprised. I can't remember what the percentage is for folks that are strep carriers. is it like 20? I can't remember. It's like 20% of the population carries, I think, strep pneumo in their nasal passages. And it's just nuts. But anyways, point being is you have these pathogens that just live on you and they just hang out there. Okay, now granted, when you take a shower and whatnot, that's why I use dial soap because it's antibacterial, all right, or Irish Spring. But point being is you're going to decrease that bacterial flora, that bacteria that likes to live on you. Wipe it out, but it can repopulate itself. But you're going to see all these different processes go to work here, and I just love it. So speaking of the skin, let's talk about, all right, restricting or preventing entry into our body here. So of course, it starts off here with the cutaneous membrane. And literally, this goes back to chapter 6, right? An epidermis, one of the characteristics of the epidermis is that it's epithelial cells, right? What's one of the characteristics of epithelial cells besides the fact that it's avascular? Epithelium is avascular, means it has no blood supply. But what's the other characteristics? This goes back to chapter 5. Well, okay, you're thinking about the skin specifically, but it's tightly packed. The cells are tightly packed. So that physical barrier, when those cells are tightly packed, and it kind of goes into the whole, Sam, the keratinization there, when the cells, because those keratinocytes, when they migrate out of the stratum basale into the stratum spinosum, one of the first things they do is start to form physical bonds, all right, desmosomes. They start to create these intracellular adhesions. So they become tightly packed, but they also make it difficult for things to slip in between the cells. So it's a perfect kind of situation when we're discussing, right, how great a physical barrier is, specifically the epidermis there. The epidermis. Okay, so what else are we going to see in our skin? All right, I don't know how much you guys talked about it in 210, but you've got glands throughout your skin. Apocrine glands, sebaceous glands, all right, and merocrine glands. Sebaceous glands are a gland kind of that are holocrine, the self-destructive gland. And so those glands specifically, right, will produce sebum. Sebum is antimicrobial. So that's why I'd always kind of, I won't say makes me laugh, but when a patient gets off after I'm examining them and they're laying on the tissue paper there, and there'll be a lot of oil on the tissue paper. And people always remark about that. And they're like, oh my gosh, that's awful. I said, no, that's good. Because that comes from the sebum, which is antimicrobial, and it's helping to prevent microorganisms from populating your skin surface. So your sebaceous glands and your sweat glands are going to produce, right, all of these different antimicrobial compounds, right? Lysozyme, all right, which is going to be also, if you recall in chapter 16, that's a big component of your tears. So your tears produce lysozyme and defensins there. And so both of those have an antimicrobial characteristic there. All right, our mucous membranes. Like I said, they're going to be lining the entrances and exits to your body. And so they're going to produce that mucus there, and itself is going to release those antimicrobial substances, and there's that IgA. That is your antibody A. And that's in high concentrations in your mucous membranes there. And then again, what do we have more of? Defenses, lysozyme. So it seems like your body has these kind of backup systems. It's not in short supply of making things, right, that are going to help protect you and keep you safe. All right? I got to take a sip. I'm talking too fast. Am I talking fast? Willie thinks I am. It's what happens when I get coffee in my body.
Feels good.
It does feel good. This is my third cup. I would have another one, but my wife would kill me. All right. So just kind of keep that in mind, right? These areas here, all right, just for a few, our two membranes there, the cutaneous membrane and our mucous membranes are going to have all these secretions and productions of material that are going to help combat, right, our immune system, not combat the immune system, but combat pathogens. So we'll see in other areas here. Look at all these different places that we'll see, right, more components that are created to help, right, with the immune response. And our respiratory tract, you learned about that way back in chapter 5, right? You have that pseudostratified ciliated columnar epithelium that's producing, all right, the goblet cells are producing the mucin, which makes the mucus, along with a bunch of the glands there. But the cilia help to move anything that gets trapped in the mucus towards your trachea, towards your pharynx there. It's not going to push it down into the lungs. It's going to sweep everything up towards your throat, right? GI tract, first thing, your saliva, right? You have two important enzymes there, right, that are created for digestion, okay? But, right, also in your saliva, you are going to be producing, right, these types of substances that are antimicrobial, right? But the best thing in my mind is this stuff. Booyah. Stomach acid. If you can make it past stomach acid, right, you are one tough cookie. H. pylori is the bacteria that can live in an acidic environment. I just had a patient that's been combating this bacteria. It's called C. difficile. Have you guys learned about that? Yeah. Yeah. And if you know anything about antibiotics, I mean, when you're taking vancomycin, which is like one of the hardest hitting type of antibiotics, and you got to be on it for a while, I think C. difficile means really difficult to kill because it is tough and it likes to reside there in the GI tract. So it's a really pain in the rear end. All right, so what we call commensal microflora. This is the stuff that lives on your skin. Okay, the nice thing is, as long as it stays on the outside of your skin, all right, or inside your digestive system, like in your intestines, right, where it's supposed to be, it's non-pathogenic. But some of these microflora, these pathogens, are what we call opportunistic organisms, which means if you grant them entry into your bloodstream or to some more sensitive areas there, they are going to try to do what they do. And they're going to try to, I'm not going to say that their mission is to kill you, all right, but as a result of that, they can cause a lot of issues there. So these are what we refer to as opportunistic or potentially pathogenic organisms if they are able to gain entry into places where they're not supposed to be, right? And so we definitely don't want some of those organisms going into your bloodstream because then you can go septic. That's why they always are worried about puncture wounds into your digestive system, especially your large intestine, for that specific reason, because that normal microflora can gain access to your bloodstream and then gain access to more sensitive areas there. All right, so let's move past. So we just did, I'm going to come right back to this. Whoops. We just did, all right, skin and mucosal membranes there. All right, prevention of entry. Now let's get into our non-specific, which are going to be cells. We're going to talk about some of the roles of certain cells, and then our chemicals that are involved, and then our physiological responses there. Okay, like inflammation and fever. All right, so let's talk about some of the internal cells there. One of the big characteristics when we're talking about cells that are part of the innate immune system is they have to have, all right, well, they don't have to, but it's very helpful if they have this special ability. What does that mean? Phagocytic cells. Yeah, that's right. Cell eating, Pac-Man. They go around and gobble things up. And so these three cells listed up here, the neutrophils, the macrophages, and the dendritic cells, that's what they're going to do. So they're going to completely engulf unwanted substances by eating it. Seems pretty decent to me because inside of these cells, they are going to have specific organelles, like lysosomes, for example, that are going to kick the crap out of whatever that unwanted substance was. It could be a cell debris, right? It could actually be a bacterium, right? Or a virus. And so what they'll do is they'll digest it. They'll actually gobble it up. So when we're talking about our neutrophils and macrophages here, right, these cells have that specific lysosome that what's going to happen is it's going to create, if you recall, well, I'll show you a picture. What am I doing? Let me just show you the picture. So here's your infectious agent. And here comes one of those three types of cells. Our example talks about the macrophage. So it gobbles up that infectious agent and it packages it into a phagosome. So it's isolated from the internal environment of the cell. Not bad, okay? We've managed to, pretty much handicap this infectious agent, but we want to destroy it. So what are we going to do? We're going to bind it with our lysosome, which is filled with digestive enzymes, right? And we're going to combine the two. And then the digestive enzymes inside that lysosome, once it combines with the phagosome, which we now call a phagolysosome, those digestive enzymes are going to just gobble up and melt away that infectious agent. And then just like everything does after it eats, it's going to poop. And then it's just going to spit out the remains of that infectious agent. Boom. Okay. So this is one of the ways that we're able to get rid of an infectious agent. And it's nonspecific. So that macrophage or that neutrophil, it will scan or it will analyze, give it an ocular pat down, that infectious agent, and then it'll identify it, because that's what usually these cells will do. They're going to look at something, cells or antigens, which are protein or sugar solutes, I guess not solutes, that's a horrible word to use, substances. And they're going to say, is this part of my body or is it not part of my body? Is it self or is it non-self? If it's self, usually leaves it alone. If it's non-self, then it's going to take it out. And we'll talk about how that all works. This process here, the respiratory burst, this stuff is awesome. Has anyone ever heard of antioxidants? They sell them in like vitamin stores all the time. They say they're great for you, all right? Because, all right, oxidants can cause damage to cells and tissues and whatnot. And I think I've used the example before with y'all, right? Imagine a kid with a yo-yo, all right, in a China shop just swinging it around. That yo-yo just starts blasting through things. So that's what an oxidant is. It has what we call free radicals, okay? And those free radicals have one electron that swings around in that valence shell, and it just tears through things. So antioxidants take that out. And so this respiratory burst, that's the process, right? So if you've ever heard of the enzyme superoxide dismutase, that's an enzyme that is utilized in the respiratory burst here. So a lot of people will take superoxide dismutase because it will help deal with specifically our reactive oxygen containing molecules there, some of those free radicals there. All right. So then how do we get rid of all the garbage inside the macrophage or the neutrophil that took it in? We just poop it out. Exocytosis there. All right. So here are dendritic cells, and this is a good kind of lead in to the role of antigen presenting cells, otherwise known as APCs. Dendritic cell is an example of that. Macrophages also have the APC. We'll talk about that in a little bit. But dendritic cells, all right, they destroy, like on this picture here, so they're going to destroy this infectious agent, and then it's going to do something kind of weird. I say it's weird, right? Then it's going to take the remains of that infectious agent and it's going to put it all over its body. All right. And that's what it does. And we'll talk about the reason why it does that, because later on, it's going to go up to another immune cell, right? One of your T lymphocytes, and it's going to display the parts of that infectious agent to the immune cell and say, hey, look, I came across this. What is this? And so obviously, if it's an infectious agent, it's a foreign material, so we can initiate the immune response. So we'll talk about that process. So basically, dendritic cells, macrophages can do this too. They'll digest whatever that component is, and then they'll just display parts of that thing on the plasma membrane, okay? So that's one of the things, oops, going the wrong way. Come on, guys. All right, that's one of the things that is important here. So keep in mind, right, when dendritic cells destroy those particles, they're going to present those particles, right, to one of our T lymphocytes. And then most likely that will initiate the immune response. So we need this to happen, right, when we want to start our adaptive immunity here. And so macrophages can do the same thing. So far, any questions? If I'm moving too fast, let me know. No? Good? Okay. It's not in the outline? Good thing you came to class then. You learn so much with me, right? Plus it's fun, right? All right. So let's talk about some of the other cells here when we are discussing our nonspecific internal defenses here. So now we're going to deal with two cells here, and we've talked about them a little bit, but now we're going to get into a little bit more detail, our pro-inflammatory cells. So these cells are going to help with the inflammation process there. So basophils and mast cells are going to help with inflammation. Right? So one's in the blood, one's in the tissue. Basophils got the blood covered, mast cells got the tissue covered. Good on that? Okay. All right, perfect. So what we're going to see is these cells are going to release from the granules, because remember, basophils are granulocyte. And so they're going to release these chemicals here, right, that are going to allow fluid to move from blood to any injured tissue. So like when you scrape your knee, and I think the example that your book uses is somebody skateboarding, they fall off the skateboard, they skin their knee. And so in that case, we need to rush some of our immune cells to the site of injury. The mast cells help with that because they're going to release the heparin. I already talked about this. And they're going to release the histamine, making your blood cells leaky and allowing stuff to flow out of your blood into the tissue there. And so they're very, very helpful. At the same time, too, they're going to shoot up a signal flare. And these signal flares are called chemotaxic chemicals. This is how, because think about it. Have you ever wondered, like, how do the cells know where to go? How do they know where the problem is? They don't have eyes. They don't have ears. They cannot figure out where to go. So here's what's going to happen. The damaged tissue from that person skinning their knee, all right, is going to release chemicals. And those chemicals will attract our immune cells to the site of injury. Also, some of the cells that have already leaked out of the blood and some of the cells that are in the tissue that might not have been damaged, they're going to start to release these chemotaxic chemicals here. And so they're going to attract the immune cells to the area or the site of tissue damage. Like I tell folks, it's like a signal flare, okay? You're stuck on a deserted island, planes going overhead at night, shoot the signal flare off, they'll see the signal flare, they'll come and rescue you. That's what's going to happen here with your immune cells here. So keep in mind, both of these cells, and I feel like everybody in this room knows this because we talked about it already, are going to release these two chemicals. One in the tissue and one in the blood. Okay, just make sure that you don't confuse, all right, those chemicals with what they do. All right, histamine makes your blood vessels leaky, and then our heparin prevents the blood from clotting. Right, because we don't want to clot those leaky blood vessels up yet. We want to get stuff from the blood into the tissues there. The mast cell. Oh, no. Oh, you're saying which one of those chemicals? Yeah, okay. So it's going to be.
Which one's in the tissues?
Oh, okay. I see what you're saying. Which of those chemicals is really? It's going to be on both sides, because you'll have the the basophils that are releasing it from the blood vessel side, the mast cells, which are going to be releasing it from the tissue side.
Both release them.
Yeah.
Blood and
Yes, both basophils and mast cells are going to release that. Yep. All right. And then, of course, don't forget about these guys, y'all. Eicosanoids. Those are those locally acting hormones. You remember those, right? Prostaglandins, prostacyclines, leukotrienes. All right. Those are going to come from, and if you think about it, it makes sense. When in that example where the kid skins his knee, you've damaged cells, right? You've damaged the plasma membrane, which means you've damaged, all right, the plasma membrane, which is going to cause arachidonic acid, okay, which is going to be fatty acid that sits in the plasma membrane of every single cell. And it's going to start to break down. And during that breaking down process, that enzymatic cascade, as we call it, it's going to make, all right, what's going to make the eicosanoids, but prostaglandins, prostacyclines. So those also will increase inflammation. That's why you take aspirin, right? Aspirin is going to block, right, I think it's the COX enzyme. I don't know if it's COX 1 and 2, but if anyone's familiar with the enzymatic cascade, it blocks certain enzymes, right, from making certain eicosanoids there. Helps decrease inflammation. All right, so here's a perfect example. Take a look at that picture. Here you can see a pro-inflammatory cell, the basophil, releasing its chemicals onto the blood vessels there. Right? And so it's going to cause anticoagulation, and it's going to make your blood vessels leaky. And all of that is going to cause fluid then to leak out along with plasma proteins, right? What do plasma proteins do? What are they responsible for? We just took a test on it, so I know every single one of you knows this somewhere. What do you think? Plasma proteins? Nailed it. Yeah, they're going to be responsible for the colloid, oncotic, or osmotic pressure. So think about it. Your blood vessels have become leaky. So the plasma proteins now leak out of your blood and into the tissues. So what's going to follow it? Fluid. It's just going to, by design. And so what happens when fluid leaves your blood vessels and into your tissues? What happens to the tissues? They swell up. Inflammation. Booyah. Okay? So that's what we're seeing here. All right, keep in mind, please, please, please don't forget these two cells are responsible for that type of scenario that we just talked about. Cool, not so bad? Not too bad? All right, now we're going to talk about one of my favorite kind of cells. I just think the name's cool. Not the macrophage yet. Yeah, that's probably, that could quite possibly be my favorite, but I like this one because look at the name. It's cool. All right, natural killer cells. So does anyone know what apoptosis is? Nailed it. Okay, good. Cell death. So these cells do exactly what their name says they do. They kill cells. What type of cells? Bad cells. Cells that we don't want hanging around. Okay, unhealthy or unwanted cells here. All right, so these cells, like any other type, right, are formed in the bone marrow. They come from the same cell line, right, as your lymphocytes do. All right, but here's what happens. They get made in the bone marrow. First thing that they do is get dropped off into the bloodstream, and then they circulate throughout the body. And they're basically just going around looking for a home. So once they find their home, they're going to go and stay in their home, which is a secondary lymphatic structure. What's an example of a secondary lymphatic structure? Tonsils, perfect. Lymph nodes, all right, spleen. All of those are secondary lymphatic structures. So they're going to take residence, even maybe in MALT, right, in the mucosal-associated lymphatic tissue. So they're just going to go and hang out there. This is what I love about these cells. So they're going to undergo this process called immune surveillance. I love it. Sounds cool. They just decide to go for a walkabout. So they could be living in their lymph node, and then one day, you know what, I'm just going to go out and take a stroll through the bloodstream. And that's what they do. They get up and they just start to circulate throughout. And that's what they literally do. They patrol the body and they just evaluate everything. So they're going to analyze the cells, right? And they're going to find which cells are self, which are part of you, and which cells are not a part of you. And if they come across a non-self cell, then they could potentially initiate the immune response, wipe it out. Question?
Yes, sorry. Are these cells when you have like an autoimmune disease? Are they involved in that?
They can mostly it's going to be your lymphocytes that are usually involved in that. Yeah, yeah. For these here, it's possible. I don't know really the answer to that question. No, it's usually your lymphocytes that are going to play a role in that. Okay, so they got a great job. Look for bad cells there. And so they like to go after virus-infected cells, bacterial infector cells, big time with tumor cells. They do play a role in transplanted tissue. So, of course, when you have a transplant of some sort, right, liver, kidney, you have to take immunosuppressant drugs because these, when you get tissue from somebody else, that's foreign tissue. That tissue does not contain your DNA. We're going to learn about this process. Your cells on a regular basis, healthy cells, are literally going to start producing these proteins inside the cell. And then your healthy cells will start displaying, okay, these protein fragments on their plasma membrane. So it reflects your own DNA. So one of these cells here will come along, T lymphocytes, they have their own class 2 that comes along, and they're just doing their check. They're patrolling, they're checking, right? And as long as these cells, your healthy, normal self cells, are displaying your normal DNA protein fragments, you're good. They leave it alone, right? It's usually when they start to display abnormal protein fragments, maybe from a virus or a fungus or a parasitic worm or even a bacteria. That's when we have a different scenario here, right? So transplanted tissue, your body, all right, produces its normal proteins that it recognizes because of the DNA, all right, structure. And then transplanted tissue, though, it's not your DNA, which means it's going to form, or it's going to display abnormal tissue or abnormal proteins, which these cells can identify as non-self and initiate the immune response, which we don't want that to happen. That's why these folks have to take, right, these immunosuppressant drugs there. So how do they do it? I love it. They release chemicals. And you want to know this because we're going to talk about some of our other cells when we get to the cell-mediated immunity here that do something very similar. Some of your T lymphocytes do the same thing. So this is cool. So natural killer cells, when they interact with, right, an unhealthy cell, okay, maybe the cell has started the process of becoming cancerous, right? So the first thing it does is it starts to punch holes into that unhealthy cell by releasing, and I love, this is one of the few times where I'm really appreciative of scientists because they actually name something, all right, after what it does. So perforins are going to perforate the membrane, the plasma membrane. It's going to create a hole in it, okay? So it punches a hole in there, hopefully more than one, and then it's going to pump in, right, this stuff here called granzymes. And these are these chemicals that are essentially going to liquidate the inside of the cell. So they're going to pour in these granzymes, and it's going to cause that cell to die. Pretty much, it's going to just cause it to shrivel right up. I'll show you a picture. I'll come right back to this. There it is. There's the beauty of the natural killer cell. It's found in unwanted or an unhealthy cell. And so it starts to release the perforins, and it punches a hole into the plasma membrane. And then next, it just starts pumping in the granzymes. The granzymes just eat away and destroy the internal structures of that cell, wipe it out, kill it. Ends up apoptosis. And that's what natural killer cells do. I think that's pretty basic. And so we're going to talk more about some of the other cells later on. Questions so far? I feel like I'm going fast, but we're only 16 slides in, so I guess I'm going slow. Has anyone ever tried Black Rifle coffee? It's tasty. Yeah, I mean, just black is my favorite. I don't drink it just black, but I like it. Okay, so let's talk about our next type of internal defense cell, and that's going to be the eosinophils. Now, we talked a little bit about these in chapter 18. These ones here, you're going to see elevated in allergic reactions, but also we're going to see them in what we call multicellular parasites or parasitic worm infections. So, of course, if a patient comes in and they have an elevated eosinophil count, and then you're taking their history, hey, have you traveled outside the country? Yes, I was down in the Amazon for a while. Then that's one of the first things you should be thinking. All right, this person probably picked up a parasite down there, and that's why they have an elevated eosinophil count. Same type of scenario here. So the eosinophils, remember, they're a granulocyte. So those little granules inside, when we look at these cells, I'll show you a picture here. See all those little red balls there or dots? Okay, those are the granules. And so those are going to contain these cytotoxic chemicals. Same type of scenario. Those cytotoxic chemicals are going to be released, and they're going to start to attack whatever that pathogen is. Whoops, went the wrong way. Right? Which are usually going to be, all right, worm infections. So the same type of scenario, we're going to start punching holes through, all right, the plasma membrane or the outer surface of those parasites there. And bam. Keep in mind, we will see them elevated. Eosinophils are going to be elevated in allergic reactions, but also asthmatics. Because a lot of times, I shouldn't say a lot of times, but there is a significant number of asthma attacks or asthma conditions that are allergy related. Right? A lot of folks will complain of having to use their inhaler more often or having asthma attacks during allergy season, or if they're living with an allergen like what I have to live with cats because I'm allergic to cats. Yeah, I love cats though. Hey y'all, I just want to let you know I'm getting a new dog soon. Puppy, Rottweiler. Love Rotties. Pardon? One of them, yeah. That was my old Rottie. She was a great dog. I'll miss her every day. Pardon? Oh, Rotties are stubborn. Yes, yes. You have to be. I'm not looking forward to getting up at 4 a.m. and taking the dog outside to pee. I'll let my wife do that. Yeah. Well, I'm hoping we're taking care of my brother-in-law's dog, and his dog hates other animals, loves people, but hates other animals. You cannot bring a dog near. So we have to wait another week until we get rid of the dog so we can get our dog. But yeah. Why would I just wouldn't want to be the owner of a dog that doesn't like other dogs, you know? I can see that I definitely won't be an owner of a dog that doesn't like people. Okay, so we'll talk later on about how eosinophils deal with the antigen-antibody complex. But an antigen-antibody complex is an antigen is a foreign protein or carbohydrate, usually a large polysaccharide. And antigens are going to be a protein that are going to essentially kind of cover up. Right, the antibodies will cover up the antigen and kind of render it useless. Have y'all ever seen, I don't remember which Matrix movie it was, the second one? I think when Neo's, has anyone ever seen the Matrix trilogy or anything? Any of those movies? Jeez. All right, this is not going to be a very good example then. I'm trying to think of a good pile on. All right, well, anyways, here's basically what happens. An antigen is present, antibodies cover it up so it renders it useless, so it can't affect anything. Well, basically, an eosinophil comes along and just cleans up the garbage. You've just got this big clump of whatever, right, floating around, and eosinophils will come along and they'll just gobble up the antigen-antibody complexes. I'll show you a picture later on. Okay, so how does this all work? How do these cells know if the pathogen or whatever it is evaluating is self or non-self? Well, they look at the cell or the structure, right, to see if the receptors on it are foreign, okay, or if they recognize it because it is, I'm not going to say domestic, but if it's part of your body there, okay? So they have to be able to recognize those receptors. If they don't recognize the receptors and they recognize them as foreign, then they're going to initiate, all right, the immune response there. All right, so keep in mind, and you learned about some of these qualities back in chapter 4 when you were learning about cell surface receptors or cell surface markers here, right? So they sit right on the surface of the cell. And so when these like natural killer cells come along or these T lymphocytes come along and they're evaluating, we'll learn this process, and they're evaluating the cell to see if it's part of us or if it's not part of us, right? They will evaluate that receptor. If they recognize it, they move on. They don't recognize it, then we have a different scenario going on. So here you can see, right, how our eosinophils will attack, right, a parasitic worm, for example. All right, let's move on now past the cells. I'm going to go back several slides here. I want to show you our progress. All right, so we're done with the cells. We're almost done with innate. Now we're going to move on here to the chemicals and then our physiological responses. Almost done with innate. I believe, or I feel like innate immunity is a lot easier than the adaptive. But you tell me after we're done with this chapter if you agree with me. Okay, so let's talk about our chemicals or the antimicrobial proteins here. All right, so obviously the name tells you exactly what these proteins do here. So one of the classes that you're going to see are called interferons. So yes, they are cytokines. They are nonspecific, right? And what they will do is they will stop the spread of cells within the pathogens. Whoops, let me reverse that. The pathogens within the cells. Okay, flip that one. Right, so what could that be? Viruses, right? They love to get inside of our cells. I told you they like to turn your cells into factories. And then we'll get bacteria that will like to jump inside the cells too. So we have two types here that we're going to talk about, actually three, right? So you got interferon alpha, interferon beta, right? And so these interferons, and this is important that you know this because we'll talk, these chemicals will be brought up again when we get into adaptive or cell-mediated immunity here. So make sure that you really, really understand this here. So the cells that make these are white blood cells. Awesome, because we want that. And then this also makes it helpful for us we have our cells that are infected by a virus. Those cells will produce these chemicals, these cytokines, which is good because they'll almost act like a signal flare of some sort there, right? And so here's what we'll see, right? When we're talking about interferon alpha and interferon beta here. So these cells will release these interferons, and they'll go to the cells that are right next door, and it helps to prevent their infection. Whoops, I'll show you a picture here in a second. All right, so how do they do this? What they'll do is, when your viruses are inside of your cells trying to reproduce and make more of themselves, they have to make certain components to the virus there. Some of those are going to be one of our macromolecules, our nucleic acids here. But a big component is they're going to prevent the viruses from being able to make components to the viral capsid, right? And which is the kind of the head structure there of the virus there. So just think of it like this. These chemicals, if you want to really simplify it, these chemicals make it difficult, if not impossible, for viruses to replicate inside of you. So we're shutting down that process, which is great. And at the same time, we're going to call our friends in, the natural killer cells, to come in and destroy these cells that were infected by the virus there. Right? So we're doing two things here. These interferons are going to, right, spread throughout the neighboring cells there and help them to prevent entry of viruses. Also, what they'll do is they'll prevent the cell that's infected from making components to make the virus. And then they'll let the natural killer cells know, hey, we have an unwanted or unhealthy cell over here, and the natural killer cells will come over and take those cells out. Now, the third interferon that we're going to talk about is interferon gamma. And so we'll talk about this later on too, but the T lymphocytes and the natural killer cells are going to make those. And so what happens? That interferon is going to call in our best friend, the macrophage. Remember, this thing does it all. It's going to call in the macrophage. It's going to be like, hey, Tony, get over here. I got a problem I need you to take care of. So Tony, the macrophage, comes in and wipes out that virus-infected cell. And that's basically, right, how useful these interferons, these chemicals are. I'll come right back to this slide because I want to show you this picture here. I love pictures. So here you can see, stepwise fashion. Here you can see we have an infected cell. So this infected cell is going to start to release interferon alpha and beta, and it's going to go to the neighboring cells, and it's going to tell these neighboring cells, hey, there's a virus in the area, okay? It's like a neighborhood watch type of thing, all right? There's an intruder in the area. Everybody be on high alert, okay? So the neighboring cells are going to start to make, all right, these chemicals that are going to prevent any viruses from replicating inside themselves if they get infected, right? And then the alpha and beta interferon are going to attract natural killer cells to come in and take out that unhealthy cell. So it comes in, wipes out that unhealthy cell, right? How does it do it? You know what it does. Perforins and granzymes, all right? It's going to punch holes in that unhealthy cell, and it is also going to then wipe out and liquidate the internal structures there. All right. Questions about that? Nothing? OBKB. Bill Cosby. OBKB. Can I not talk about Bill Cosby anymore? Probably not. My bad. My bad. All right. I grew up watching The Cosby Show. And Fat Albert. Has anyone ever seen Fat Albert? Yeah. That was a horrible Fat Albert, by the way. All right. Here we go. So let's talk about our next antimicrobial protein group called the complement system. Now, we could do a lot on the complement system. I'm only going to give you the most basic parts of the complement system because there's a lot of information on the complement system. So, I mean, just look at this. Over 30 plasma proteins, all right, are part of the complement system. And this is how creative we are. We just label those proteins with the letter for complement, C, and then the order of which we discovered those different proteins. So it's 1, 2, 3, all right, all those different ones. I think they should. It'd be easier, all right? instead of these weird, crazy things. So where does complement come from? You guessed it, the liver. And so complement, much like angiotensinogen, is made all the time, and it's released in the inactive form here. So again, it's just constantly on hand, and we'll use it, all right, in a case of emergency, break the glass. And that's what we'll do. And when we do, we're going to see this enzymatic cascade occur like we saw with inflammation. So one will activate another, which will activate another and cause the desired outcome, which we'll talk about here in a second. So there's two different what we call pathways that complement can operate by, right? One's called the classical pathway, then the other one is the alternative pathway. I know, real original, but basically, right, keeping it simple. When we're talking about the classical pathway, we're going to be dealing with our antibodies, right? When we're talking about the alternative pathway, we're going to see how the complement is going to bind onto a large sugar, our polysaccharides, which are usually going to be found within the bacterial and fungus walls of our pathogens. So if you've taken micro, you know what I'm talking about, right? Have y'all covered that at all in micro? Have you guys talked about the bacterial wall? Maybe not so much. Okay. I can never remember where they talk about. I think they talk about it pretty early on. It's been years since I've done micro, so you have to forgive me, okay? All right. So the nice thing about complement is that it's great for bacterial infections. I mean, it's pretty awesome. So essentially, when the complement's activated, and we'll talk about how it works, all right? Because there's a couple different ways that complement is actually, once it's activated, how it affects things, all right? So we're going to talk about those right now. But I just want you to understand, complement's similar to angiotensinogen. It's inactive. It's being produced as we're sitting here right now. Your liver's pumping complement out. So in case you're dealing with a pathogen of some sort, okay, we'll activate that complement, and then we'll talk about its effects right here on the next slide. Before I do, I'm going to take a sip. All right, here we go. The four things that can happen, and I'm going to do one of those things, then I'm going to show you a picture, and then I'll come back and do the next one. First one is inflammation. Easy. We're all familiar with it. So inflammation is only going to be enhanced by the complement. So that means it's going to trigger our mast cells and basophils to release heparin and histamine to make the cells leaky. And at the same time, it's going to send up its signal flare, and it's going to tell the most numerous type of white blood cell, the neutrophils, and their henchmen, the macrophages, to come to this site. So it's going to look something like this. Boom. So here's the problem area, okay? And so the complement is then going to tell the mast cells and the basophils to release their chemicals, right, to make the blood cells leaky and to prevent clotting. So these fellas here can make it to the tissue, squeeze out of the blood cell and get, squeeze out of the blood vessel and get into the tissue here, right? And then they'll also going to send up their signal flares here, boo, 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 and they're going to shoot it up there, and it's going to attract, all right, these white blood cells to the area, right? So it's going to compound, or, you know, and I think it's aptly named complement. It's going to complement inflammation. It's going to enhance that inflammation there. All right, so that's the first way that complement can work. The next one is called opsonization. All right, and so opsonization, we're actually going to see, all right, a binding to the pathogen here. So essentially what we're going to do is we're almost going to put a label on something, right? I'm sure that if you've ever gone to, I like to consider myself thrifty. Some people would call me cheap, whatever you want to call me, all right? But like, I bought a refrigerator at Lowe's for a ridiculously cheap price because it had this big scratch on the side. And they put a big tag on it, and it said scratch and dent, right? Which drew me over there because it was this big sign, right? And guess where that dented side of my refrigerator is facing? The wall, okay? Smart shopping. Well, guess what? All right, when we're talking about complement, right? When we talk about opsonization, that's what happens. So here's what happens. You've got this pathogen that looks like a bacteria to me. The complement attaches itself to the pathogen. And it's saying, it's like putting its arms up to white blood cells, to phagocytic cells. Hey, I got something over here. And that's what's going to happen. The macrophage will come right over there and gobble the whole thing up. So that's opsonization. It makes something more noticeable. And so that's what we're going to see here, right? So now we can phagocytize the cell, take that thing out. The next one is cytolysis. What does cyto mean? Cell, nailed it. And then lysing, we're going to chop it up, okay? So here's where we're going to see, I love this one. This is probably, we used to call it when I was learning about this in med school, they call it the membrane attack complex, the MAC. We used to call it the Mac attack, all right? Big Mac, all right? So that's essentially what we're going to see here. With cytolysis, we're going to see the complement's going to come along, and it is going to do something similar to what the natural killer cells did, right? When they were dealing with their pathogens. It's going to come along, and it's going to start punching holes into that pathogen's membrane there. And when I say holes, it's going to be multiple holes. So eventually it compromises the structure there. Fluid enters in, fills it up, pop, like a water balloon that you filled up with too much water. Right? So that's what we're going to see here on this slide, cytolysis. So you can see, here's our pathogen, all right? And then the complement starts to build these holes inside of it. And then water floods, I say water, but fluid, depending on where this is, mostly water, will flood into that cell and cause it to burst. Okay, Big Mac attack, bam. All right, so that's the third way that complement is going to handle our pathogens. And then the last one is, you heard me mention antigen, antibody, immune complexes, right? So here we're going to see right? Complement is going to attach onto the antigen antibody complex. So you'll understand it a little bit better when we get into the plasma protein, not the plasma proteins, when we get into the plasma cells and we talk about antibodies that they produce, and we'll talk about how it does. So for right now, don't worry if you don't really quite understand this, right? It's not too complicated, but what we're going to see is, think of the complement as like a link, all right? And so the complement is going to attach itself to an antibody and that antibody is attached itself to an antigen, and then that whole thing is going to attach itself to a red blood cell. So the red blood cell is like a sea turtle with barnacles. You ever seen a sea turtle with barnacles attached to it? It's just going to go along for a ride, right? And so here's what's happening. Think of the red blood cell as the sea turtle, and then the barnacles attach onto it. Well, the complement is going to attach onto the red blood cell, dragging behind it the antibody antigen complex, and it's going to head to one of our two favorite organs here, the liver and the spleen, where inside those organs, specifically the spleen, we're going to strip that stuff off of the red blood cell, and then there's macrophages in there that will crush it. So, oops. Let me show you. Right here. Booyah. So here you can see, here's our antigen. Here's our antibody that's attached to the antigen, making the antigen relatively useless, all right, and inert. And then the complement comes along, attaches on to the antibody, and then the complement will attach onto the erythrocyte. Erythrocytes have to go to the liver and spleen, all right? They eventually wind up there. So when they go to those structures, they're going to wind up encountering a white blood cell of some sort, right? And so they'll strip off this part here, and usually that gets phagocytized. Just random. Yeah, it just pulls it in. Yeah. Oh, yeah. Yeah, yeah. Well, it's possible. I don't know if it does, but yeah. No, because 120 days is a long time, so it'll make it. Yeah. No. I see what you're saying, because I used to think that, because the liver and the spleen are where our red blood cells go to die and get recycled. But it just happens. They flow through there all the time. And so it gets, Yep.
Doesn't necessarily mean the red blood cells are death.
Right, exactly. Yep, it's not marked for death or anything like that. Yeah, the red blood cell is just random. It's just, it could be a 2-day-old red blood cell. It could be a 100-day-old red blood cell.
It's the liver, spleen, that fat phase.
Yep, we'll strip this off. Yep, strip it off, and then the red blood cell can go on its way. It's like, wait a minute, you're 121 days, you're gone, buddy. Okay, so questions on the complement. All right, let me check the time here. All right, this is a good spot to stop